The human papillomavirus has an outer shell or capsid, comprised of the major capsid protein L1 and the minor capsid protein L2. It is the L1 protein that stimulates the major antibody response in humans. Within the HPV capsid is the viral DNA. Gardasil is made of virus-like vaccine particles, bioengineered from only L1 proteins. These vaccine particles are empty shells containing no viral DNA and are therefore not capable of causing infection or HPV-related diseases. However, because they are made of L1 protein, the vaccine particles are able to mimic the virus and elicit an antibody response. In creating the vaccine, purified L1 proteins produced in yeast cells come together to form pentamers, which then self-assemble to form empty particles that closely resemble the HPV particle. Gardasil contains 4 vaccine particle types, each composed of L1 proteins of either HPV type 6, 11, 16, or 18. A proprietary adjuvant is added to enhance immunogenicity. When injected intramuscularly, the vaccine particles are recognized as foreign by a variety of cells, including antigen-presenting cells, APCs, which engulf and process them. The APCs then travel through the lymphatic system to lymph nodes. Here, the antigen is presented to naive T helper cells, triggering T cell activation. B cells also process the vaccine particles, and when they subsequently encounter T helper cells, cytokines stimulate their differentiation into plasma cells and memory B cells. The plasma cells secrete high levels of antibodies specific to the HPV types targeted by vaccine. Some of these antibodies are capable of neutralizing HPV. Leaving the lymph nodes, antibodies travel in the bloodstream throughout the body. From the bloodstream, neutralizing antibodies transudate into genital epithelium, and some antibodies pass into cervical secretions. In clinical trials, Gardasil provided high efficacy against HPV 16 and 18-related SIN2-3 and AIS in women 16 to 26 years old and naive to the relevant HPV vaccine types. Gardasil was also highly efficacious in clinical trials against HPV 6, 11, 16, and 18-related external genital lesions, including vulvar and vaginal intraepithelial neoplasia and genital warts, and vaccinated individuals who received a challenge dose of Gardasil 5 years after vaccination exhibited an immune memory response for each of the 4 HPV types in Gardasil. Additionally, no cases of SIN2-3 due to waning immunity were observed in the patients receiving Gardasil through 5 years in the prophylactic efficacy group in an extension phase of Protocol 007. One of the hallmarks of vaccines that confer long-term immune protection is immune memory. Immune memory is one of several potential mechanisms that are initiated soon after vaccination with Gardasil. Following HPV exposure, even very low levels of existing circulating HPV antibodies may neutralize most viruses. However, some HPVs may reach the basal epithelium through areas of microtrauma where they may trigger an immune memory response. The process is initiated when antigen-presenting cells take up the virus and travel to lymph nodes. Here, the APCs initiate an immune memory response by activating memory T helper cells, which in turn activate memory B cells. Once activated, memory B cells quickly differentiate into plasma cells capable of producing larger amounts of higher affinity antibodies than in the primary response. This mechanism of immune memory results in a strong and rapid antibody response that may start 24 to 48 hours after exposure. Studies have also suggested that disruption of the genital epithelium may be required for HPVs to reach the basal cells of the squamous epithelium, which are the only cells that support productive infection. Under experimental conditions, studies have suggested that unlike most viruses, HPVs exhibit remarkably slow kinetics of infection of these cells and may therefore be susceptible to neutralizing antibodies for at least 2 days. HPV antibodies induced by Gardasil are also active in fully keratinized non-mucosal epithelium. Here, they are protective against HPV infection, which cause external genital warts, and vulvar and vaginal